Hello, everyone, and welcome to Monday Minutes. This week you have 2 lectures to view on our playlist on YouTube. The second half of Neurons, Nerves, and the Peripheral Nervous System, and the first part of the Central Nervous System, called Spinal Cord and Spinal Nerves. This edition of Monday Minutes will be about an hour long, so let's go ahead and get started. This week, we're continuing our discussion of neurons, nerves, and the peripheral nervous system, as well as the spinal cord, which is the first lecture about the central nervous system. In this second lecture about the peripheral nervous system, we learn about the process of the action potential. We learn about the physiology of conductivity. So we talked about this concept that resting membrane potential is a gradient of charge across a cell membrane. Remember, all cells have this resting membrane potential. And all that means is that there's a differential in the concentration of certain molecules on both sides of a cell membrane. That makes sense. And some of those molecules carry a charge. So there's also a difference in charge. There's a charge gradient across any plasma membrane on any cell. That's called resting membrane potential. Remember, there are 4 things that contribute the most to resting membrane potential: sodium, potassium, chloride, and protein. All cells have resting membrane potential. There are only 2 types of cells that can change that resting membrane potential into electrical current. That is the neuron and the muscle fiber. So this question of the action potential and how an action potential forms is all about changing that resting membrane potential. Remember, it's ion channels in the membrane of the neuron or the muscle fiber that are going to play a huge role here. Ion channels are simply proteins that are embedded in the membrane of the neuron that can open and close at certain voltages. We say that they are voltage gated. So when the membrane potential reaches certain values, either positive or negative, these ion channels either open or close. So we talked about this concept of depolarization, which is a state that happens when the inside of a cell, a neuron or a muscle fiber, becomes suddenly less negative. Or another way to say that is more positive. And in comparison, hyperpolarization is the state where the inside of the neuron or muscle fiber suddenly becomes more negative or less positive. That's called hyperpolarization. In the neuron, sodium channels and potassium channels are doing this work. So yes, resting membrane potential is determined primarily by concentrations of sodium, potassium, chloride, and proteins. But conductivity, the movement of an electrical current down the membrane of the neuron, is primarily regulated through sodium channels and potassium channels. Remember, sodium is normally higher outside of the neuron than inside, and potassium is the opposite. Potassium is normally higher inside the neuron than outside. So, when sodium channels open, sodium rushes in to the neuron. When potassium channels open, potassium rushes out of the neuron. And since both sodium and potassium have positive charge on them, that means that when sodium channels open and sodium rushes in, we're bringing more positive charge into the neuron. And the neuron's membrane potential is going to become less negative or more positive. That's depolarization. When potassium channels open, potassium rushes out of the cell, taking its positive charge with it. That means that membrane potential becomes less positive or more negative. That's hyperpolarization. It's really important that you understand these concepts of how these two ions move. And there's no trick I can give you to remember that sodium moves in and potassium moves out. You just have to memorize that fact. So when we're talking about the action potential, we're talking about the unit of electrical current that's traveling down the membrane of the neuron. Remember, electrical current doesn't travel through the cytoplasm of the neuron. It travels along the membrane of the neuron because that's where these gradients are, on either side of that membrane. In addition to depolarization and hyperpolarization, there are two other states that we need to understand when it comes to an action potential. Depolarization is going to make the inside of the cell more positive or less negative. That means that the resting membrane potential, which is approximately minus 60 millivolts, is going to become more positive very quickly. It's going to go from minus 60 to minus 40 to minus 20 to 0 to plus 10, plus 30, plus 40, and so on. That's depolarization. Membrane potential becomes more positive. Now, when it hits that plus 40 mark, something happens, and that something is that the sodium channels are going to close. When the sodium channels close, there's no way for any more sodium to rush into the cell. At the same time, potassium channels are going to open. So potassium ions are going to rush out of the cell. And we're going to see membrane potentials start to drop back down again, start to become more negative. Now, you might think that that's hyperpolarization, but remember, depolarization and hyperpolarization are relative to resting membrane potential. So when we go more positive than minus 60, that's depolarization. When we go more negative than minus 60, that's hyperpolarization. When we go from a high positive value, like the peak of depolarization, plus 40 millivolts, when we start to drop down, we're not going to be in a hyperpolarization phase until we go below resting membrane potential. Before that, we're in what's called a repolarization phase. So from plus 40 millivolts down to minus 60 millivolts, resting membrane potential, we are repolarizing the membrane. That's the time when potassium is rushing out of the cell. The potassium channels, like the sodium channels, are also voltage gated. In other words, they open and close based on what voltage the membrane is at. Repolarization is going to continue to minus 60 millivolts and beyond. At that point, it becomes hyperpolarization. So from minus 60 millivolts down to minus 75 millivolts, we are in a hyperpolarization phase, what some people call the undershoot. We need to get back to resting membrane potential, and that happens passively. When we hit minus 75 millivolts, the potassium channels are going to close. So now the sodium channels are closed and the potassium channels are closed. But there is some passive leakage of ions occurring across the membrane, a slow passive leakage, and that will help return us to resting membrane potential. So the phases of the action potential include depolarization, repolarization, hyperpolarization, and then return to resting membrane potential. And that's what this diagram is all about. In the lecture, we talk about how there's actually 3 different diagrams here, graphs and drawings. This top one just shows us when we're going to get a stimulation to the neuron. This middle one shows us the phases of the action potential, and this bottom one shows us the state of the sodium channels, just the sodium channels. So remember that the neuron at rest has the same resting membrane potential that all cells have, about minus 60 millivolts. On the x-axis here, we're looking at time passing by. And on the y-axis, we're looking at the membrane potential in millivolts. So we start off at rest with the pink line flat at minus 60, and then at time 0, something happens. The something that happens is a stimulus is applied. Now, depending on the type of neuron, this stimulus can be different things. But what that stimulus is going to do is it's going to make the membrane potential become more positive. And you can see that as the pink line starts to travel up from minus 60 towards 0. Now, this stimulus has to be strong enough to hit what we call threshold or threshold potential, which is approximately minus 40 millivolts. If we hit that threshold, an action potential is going to occur. If we don't hit that threshold, this is going to occur, this green line. We're going to go up a little bit, a little more positive in the membrane potential, but then we're just going to go right back down to resting membrane potential. If the threshold is hit, though, something's going to happen, something significant. Because at minus 40 millivolts, the threshold potential, the sodium channels are going to open in the neuron's membrane. Remember, sodium rushes in, and sodium has a positive charge. So suddenly the inside of the neuron becomes much more positive, and that's what we're seeing here. A sudden, rapid upward motion on the graph. The membrane potential is becoming much more positive from minus 40 up to 0 up to plus 40. That's depolarization. Now at plus 40 we hit a peak because at plus 40 the sodium channels are going to close. At plus 40 the other thing that happens is the potassium channels open. Remember, potassium rushes out of the neuron, taking its positive charge. So the inside of the cell starts to become less positive or more negative, and we start to see this downward motion in the graph. Remember, this is not hyperpolarization, this is repolarization. We have to get down to resting membrane potential and then below it to be in hyperpolarization. So this part here is hyperpolarization. The negative resting membrane potential below minus 60 millivolts. Now, the other thing they show us is the state of the sodium channels. At rest, sodium channels are closed. Sodium channels are voltage gated. So if the stimulus that's applied to the neuron brings the resting membrane potential up to a potential of minus 40, sodium channels are going to open. Now, up at this peak here, they're going to close again. The other thing that happens is important to remember, and that's this state of refractory periods. And that's what we'll talk about next. Refractory periods are simply moments of time when the neuron is going to resist being stimulated again to the point of producing a new action potential. There are two refractory periods. There's the absolute refractory period when no amount of stimulus can generate another action potential, and there's a relative refractory period when it would take an extra large stimulus to generate another action potential. During the relative refractory period, the sodium channels are starting to reactivate, but slowly and one by one, while the potassium channels are still open. This was another graph that we looked at in the lecture, helping us to visualize these time periods and phases of the action potential. You can see that the absolute refractory period occurs right around here. This is the time when sodium channels, shown in pink, are not only closed, and they're showing us that by having the walls touching each other, but they're also inactivated. We can see the lock there. Potassium channels are still open. Potassium is still rushing out of the cell, which is why membrane potential is becoming more negative during repolarization. During the relative refractory period, it's a little bit different. During the relative refractory period, sodium channels are starting to open up, but they're opening up one by one, not all at once. So as you're studying, make sure that you can explain this graph of the action potential. Make sure that you understand what's happening from the time that the resting membrane gets stimulated up to threshold, to the sodium channels opening and depolarization occurring, to the sodium channels closing and inactivating, and the potassium channels opening. That's repolarization. Remember, the potassium channels stay open past resting membrane potential. Now we're into hyperpolarization, all the way down to minus 75 millivolts when the potassium channels close. But neurons can't just generate one action potential. Neurons have to generate multiple action potentials, one after the other. And that's where we get into transmitting this electrical signal. The neuron has to send the signal all along its axon to the next neuron, or to the spinal cord, or some effector like a muscle or a gland. Remember that action potentials can spread in two ways. They can spread passively, and they can be propagated. And depending on whether or not the axon that we're talking about is myelinated or not, sometimes propagation is saltatory or sort of jumping. Passive spread is simply what happens to the action potential as it spreads out from the site where it was generated. The action potential is going to lose strength as it gets farther and farther from that stimulus point. Propagation occurs when that action potential gets regenerated from point to point along the membrane. We looked at this slide to sort of visualize how these electrical signals are getting transmitted. We've got a drawing of a sort of generic neuron up here, and where the red mark is, that shows us where this neuron was originally stimulated up to threshold. An action potential was generated, and it started to spread out in all directions, and that's what the darker color is showing us. Notice that it started to spread backwards up the dendrites, and it started to spread forwards into the neuron cell body. It just spreads in all directions, passively though, losing strength as it gets farther and farther away from the original stimulus. Now, if the signal is powerful enough to reach this region called the axon hillock, then it can get propagated. There are lots and lots of sodium channels sort of clustered in the axon hillock. So as long as there's enough strength in that action potential to hit the hillock, something amazing is going to happen. Sodium channels are going to open. Sodium's going to rush in. We're going to have a new depolarization event. You can see that action potentials get regenerated, propagated, all the way down the length of the axon. And that's important because axons are very long, or can be very long, and we need that signal to keep going. The only way to keep that signal going is to propagate it, to make it continue again and again and again. In a myelinated axon, it gets a little more complicated because of the myelin sheath. Remember, myelin is an insulator. The reason that myelin is present on the axons of certain neurons is because it insulates the axon. And just like insulation on wire helps the electricity travel without losing strength, the myelin around an axon helps the electrical signal travel along the axon without losing too much strength. So myelination is not continuous, it's discontinuous. It occurs in one spot, then there's a break. Then we get more myelin, then there's a break. These breaks are called nodes of Ranvier, and these nodes are packed with sodium channels. So at the axon hillock, action potentials get regenerated. As they travel under the myelin, they don't lose too much strength, but we still need to regenerate them, and that's going to happen at the node. Sodium channels are going to open, sodium's going to rush in, we're going to get more depolarization, another action potential gets generated. Under the myelin to the next node. Remember, we can't start a new action potential under the myelin because the myelin is coating the membrane of the axon. It's covering it. So even if sodium channels were to open in the membrane underneath this myelin, they're blocked. They're blocked by the myelin. So the reason there are breaks, the reason these nodes of Ranvier occur, is because we need to have a spot where the sodium channels are open and sodium can rush into the neuron. Electrical signals need to travel down the axon of one neuron so that they can then be sent to the next structure, whether it's another neuron, whether it's the spinal cord or the brain, or whether it's some effector out in the periphery, something like a muscle or a gland. The points where a neuron comes in contact with the next structure are called synapses. In the lecture, we described the presynaptic cell and the postsynaptic cell. In other words, the first cell, the one that comes to the synapse, and then the second cell, the one that's after the synapse. Presynaptic and postsynaptic. There are two kinds of synapses that we discussed. There are electrical synapses and there are chemical synapses. Remember, electrical synapses are synapses where the connection is simply gap junctions between the two membranes of that presynaptic and postsynaptic cell. So you literally get tunnels created that allow that electrical signal to travel down the neuron and very quickly and very efficiently go into the next postsynaptic cell membrane. We said electrical synapses allow for cell-to-cell transmission with virtually no delay. And that is going to happen where speed is essential. The best example of that is in cardiac cells or cardiomyocytes. We looked at this drawing of an electrical synapse. You can see a terminal bulb here at the end of the presynaptic neuron. And then snuggled up right next to it is the membrane of whatever cell is the postsynaptic cell. These two membranes are very close together, and they are literally connected with a tunnel, and that tunnel is created with gap junctions. So the electrical signal comes down the presynaptic membrane, jumps through these tunnels and into the postsynaptic membrane. Remember, the only drawback to the electrical synapse is that it's an all or none. In other words, whatever the strength is of the electrical signal that is coming down the presynaptic neuron, whatever the strength of that signal is, it's going to be transmitted to the postsynaptic cell in the exact same strength. And again, there are times in the body when that's fine, when that works perfectly. And the heart is one such example. But there are other places in the nervous system where that is not acceptable, where that doesn't work very well, because we need to start doing some fine-tuning of the electrical signal. Whatever's coming down the presynaptic neuron, we want to modify it a little bit in the postsynaptic cell. And that's where the chemical synapse comes in. Chemical synapses are much wider than electrical synapses are, relatively speaking. Chemical synapses use a chemical called a neurotransmitter to send the signal from the presynaptic cell to the postsynaptic cell. That chemical gets released from the terminal bulb inside the axon and then travels across that synapse where it then binds to receptors on the postsynaptic cell. And that's what they're showing us here in this blown up image. Neurotransmitters are stored in membrane-bound vesicles inside those terminal bulbs inside of an axon. And when the electrical signal gets down to that terminal branch of the axon, the electrical signal is going to cause calcium channels to open up. We talked about this when we discussed muscle. The calcium is going to cause these vesicles to fuse with the presynaptic cell membrane, the neuron membrane, and the chemical inside, the neurotransmitter, will get released into the synapse. It travels across the synapse and it binds to receptors on the postsynaptic cell. Here's where the fine-tuning comes in. Depending on the neurotransmitter, sometimes what happens on the postsynaptic cell is different than what was happening in terms of the electrical signal on the presynaptic side. In other words, the signal can be modified, it can be altered, it can be changed, it can be enhanced, or it can be dampened down when you have a chemical synapse. We discussed this concept of integration in the lecture. Remember, even though we often talk about the transmission of an electrical signal from one neuron to the next neuron in a very simplistic way, in reality, neurons aren't receiving a single signal at any one time. They are receiving multiple signals all the time. You can see in this drawing that this neuron cell body is receiving signals that are sort of green colored. Those are excitatory signals, as well as signals that are red colored. Those are inhibitory signals. So this one neuron is getting lots and lots of input from other sources. And what this neuron has to do is sum up all of that information so that it can then send along its signal, along its axon to the next cell. This is integration. This is receiving lots and lots of different kinds of information and putting together some summary of all of that that can then be sent along to the next cell. The last thing that we talked about in this neurons, nerves, and peripheral nervous system lecture was this concept that while neurons cannot be replaced in the nervous system, neurons are one of two kinds of cells that simply cannot divide once they're in place in the human body. Remember, it's neurons and muscle fibers that cannot be replaced. We did talk, however, about how nerves can be regenerated under certain circumstances. Remember what a nerve is. A nerve is a group of axons that are all bundled together. Just like ganglia are groups of neuron cell bodies that are all bundled together. We find nerves and ganglia in the peripheral nervous system. If the nerve is damaged, if the nerve, for example, gets cut or transected, the part of the nerve that's distal to the cut is going to die. But as long as the neuron cell bodies are intact, as long as the ganglia is intact, the upper portion of the nerve can regenerate. And we talked about this regeneration process step by step. Remember, anything that's distal to the injury is going to die and degenerate. The piece of nerve that's distal to the injury, any Schwann cells that are around that distal end to the nerve, they're going to regenerate. The immune system is going to come in the form of macrophages to clean up all that debris. And then up at the proximal end, the neuron cell bodies that are in the ganglia, they're going to swell up because activity is going to occur in order to repair that damaged nerve. The stump of the nerve, all those axons that were transected, the stump of the nerve is going to start to sprout processes at its distal end. And Schwann cells and the membrane of the axons at the cut end are going to form what's called a regeneration tube. Schwann cells produce growth-promoting chemicals called nerve growth factors that are going to help this sprouting occur. The regeneration tube is going to basically direct the regrowth of the axon. Remember, axons can regrow, but they only regrow slowly, about 3 to 5 millimeters per day. That regeneration tube guides those sprouted processes back to the original location where that axon originally traveled, reestablishing synaptic contact. Eventually, the ganglia will shrink back to its regular size. We looked at this drawing again to show us that at the cut end, remember, everything distal dies and degenerates and then gets cleaned up. We get these processes growing under the influence of nerve growth factor. A tube helps direct those processes where they need to go, and we end up reestablishing contact to whatever structure that axon should normally travel. Remember, though, we also talked about sometimes things go wrong here. Regeneration's not perfect, especially for motor nerves. Some nerves will connect to the wrong muscle fibers. Some nerves will never find muscle fibers and will just end. And of course, if muscle fibers never get their nervous input back, they're going to die. So nerve injury sometimes leads to, in fact, often leads to some kind of functional deficit, even after nerve regeneration occurs. Remember, damaged nerves in the central nervous system don't regenerate. This only happens in the peripheral nervous system. The second lecture this week is about the spinal cord and the spinal nerves. And the first thing we discuss in the lecture is the functions of the spinal cord. Remember, the spinal cord conducts nerve impulses. It helps conduct nerve impulses from the central nervous system to the peripheral nervous system. And it helps conduct nerve impulses from the peripheral nervous system to the central nervous system. The spinal cord, remember, is part of the central nervous system, along with the brain. The second function for the spinal cord is integration. The spinal cord plays a big role in combining all of that afferent sensory input from the peripheral nervous system and then through the spinal cord and up to the brain. The spinal cord has to integrate, it has to combine, it has to sum up all of that information before it sends it to the brain. The spinal cord is also capable of initiating a response. In other words, it can integrate the incoming information and initiate the response. These are referred to as reflexes. These are involuntary, stereotyped or stereotypical responses, as we call them. They're very fast. They don't require any conscious thought because the brain's not involved. Reflexes are often protective because they are so fast, and they help maintain homeostasis. The spinal cord often works in conjunction with the brain, but it is capable of reflex activity. We spend a lot of time in the lecture talking about the gross anatomy of the spinal cord because there's a lot of language for us to learn. Remember, the spinal cord is going to give rise to 31 pairs of spinal nerves. And the way the spinal nerves exit is through the vertebral column. we talk about how the numbering system for the vertebrae and the spinal nerves is just a little bit skewed. We see some language that is exactly the same. In other words, the spinal cord, like the vertebral column, has a cervical region, a thoracic, a lumbar, a sacral, and a coccygeal. But when you look at the numbers of spinal nerves, you're going to have essentially an extra spinal nerve compared to the number of individual vertebrae. Remember, the spinal cord is only about 18 inches long. It comes down from the brain to the level of about L1, the first lumbar vertebrae, where it widens and then tapers to a point called the conus medullaris, or the medullary cone. From that point on, what's coming off of that cord is nerves, nerve roots called the cauda equina. So from L2 down through the rest of the vertebral column, there is no spinal cord. There are only these spinal nerve roots, the cauda equina. This term, cauda equina, translates as the tail of the horse because these nerve roots look like long hairs. We looked at lots of diagrams explaining the relative anatomy between the vertebral column and the spinal nerves. Again, what's sort of off between the spinal nerves and the vertebral column is the numbering in the cervical region. And that's because the first cervical spinal nerve comes off of the spinal cord between the skull and the first cervical vertebra. And then there's another spinal nerve that comes off below the first cervical vertebra. So there are 8 cervical spinal nerves, but only 7 cervical vertebrae. In this lecture, we talk about the fibrous coverings that surround the spinal cord. These are going to be the same coverings that surround the brain when we get to the brain. From the outside in, we call these the dura mater, the arachnoid mater, and the pia mater. And we gave details about each layer and how it compared to the other structurally. We spent a good deal of time in the lecture looking at the anatomy of the spinal cord in cross-section or transverse section. We talked about where the white matter is and where the gray matter is. Those terms referring to the neuron cell bodies and their dendrites, that's gray matter, versus the axons of all the neurons, that's the white matter. So when you make a cross-section through the cord, the gray matter, the neuron cell bodies, are found to the interior. They make this sort of a butterfly shape that we describe as ventral horns and dorsal horns. In certain areas of the spinal cord, there's also lateral horns. The reason that dark matter is dark, remember, is because it contains some pigments, both melanin and a pigment called lipofuscin. The white matter surrounds the gray matter, and this is where all the axons are found. The spinal cord has mirror imagery. In other words, if we were to fold it right here in the center, we could fold this half over this half and they would match. There's a lot of vocabulary to learn when it comes to the spinal cord. The right and left sides of the gray matter are connected by what's called the commissure. And in the center of the commissure, there's a little canal that's actually lined with a type of epithelial cell called an ependymal cell. That central canal is full of cerebrospinal fluid, although it does close up in certain areas in adults. We can see 2 sort of splits along the midline of the spinal cord. There's the anterior median fissure and the posterior median sulcus. In this diagram of the Spinal cord in cross section, we can see the fissure here on the anterior surface and that sulcus here on the posterior surface. And here's that commissure connecting the gray matter between the left and right halves of the spinal cord. We also discussed how the nerves come off of the spinal cord. These are called spinal nerves. There's a ventral root and there's a dorsal root. The dorsal root has a ganglion associated with it. The ventral root does not. This ventral root contains sensory fibers, whereas the ventral root contains motor fibers. These two roots that are coming off of the spinal cord are going to combine and become a spinal nerve. And the spinal nerve is going to exit out of the vertebral column. In addition to a lot of new gross anatomy when it comes to the spinal cord, we also have a lot of microscopic anatomy to learn. We can describe both structural regions in the cord, and we can also describe functional regions, particularly in the white matter. The white matter of the spinal cord, remember, surrounds the gray matter. And there are bundles of axons that travel up and down the spinal cord. There are structural regions to this white matter, and they're called funiculi. We identified posterior, anterior, and lateral funiculi in the spinal cord. Axons that travel within each funiculus are bundled into smaller units called fascicles, very similar to the fascicles that we see in muscle and in the peripheral nervous system. So when you look at the spinal cord in cross section, you should be able to orient yourself based upon how these structures lie. We can see the gray matter in the center, and remember that's organized into a dorsal horn on the left and a dorsal horn on the right, a ventral horn on the left and a ventral horn on the right. Here's that commissure that connects them. Here's the central canal. This is the dorsal area of white matter. These are the posterior funiculi. This is ventral or anterior, so these are the anterior funiculi, one on either side of the anterior median fissure. Up here, that's the posterior median sulcus. These over here then are the lateral funiculi. Remember, these are structural regions. We can also describe these bundles of axons functionally. We don't refer to them as nerves when we're in the central nervous system. We refer to them as tracts. So we have both ascending tracts and descending tracts in the spinal cord, bundles of axons that are either carrying sensory information or carrying motor information. Ascending tracts are sensory. These are tracts that are heading up towards the brain. They are ascending. Descending tracts are motor. These are tracts or bundles of axons that are carrying motor information from the brain through the cord to go out into the peripheral nervous system. All bundled axons within a tract are going to be functioning together. Similar origin, similar destination, similar function. The other thing we talked about was how sometimes tracts undergo decussation, which means they start on one side, left or right, and they cross over to the other side of the spinal cord. We refer to two structures that are on the same side of the body as being ipsilateral, and two structures that are on opposite sides as being contralateral. This picture that we looked at really puts it all together for us so far and brings us back to the stimulus response pathway. Remember, early in the course, we talked about this pathway where some stimulus gets received by a receptor, travels up ascending or sensory neural pathways to a control center where the information gets integrated, a response is generated, and then travels out through descending or motor neural pathways to effector structures. In this picture, we can see a hand touching something, a pin, something sharp. That stimulus is going to be received either as pressure or touch or pain, depending on how hard this finger is impacting this pin. Receptors are picking up that pinprick and transmitting that information along ascending nerves because we're in the peripheral nervous system. Ascending sensory nerves are going to come in through the spinal nerves into the spinal cord. Sensory information travels through the dorsal root. Remember, once it gets into the cord, it's going to travel up ascending tracks towards the brain. The brain will integrate the information and initiate a response. The response is going to come down descending tracts. It will then exit through the ventral root of the spinal cord at the appropriate level, where it will have its effect on some effector structure, probably muscle, to help pull that hand out of the way of that pin. As we connect the structure and the function of the spinal cord, we can see sensory tracts, ascending tracts, in all of the three funiculi, the posterior, the anterior, and the lateral. But motor tracts, descending tracts, can only be found in two, anterior and lateral. If we were to take out the tracts from the spinal cord and the nerves in the peripheral nervous system that those tracts were connected to, if we were to take them out, they would be like ropes, groups of axons all joined together, the way a rope is made out of threads all joined together. But if we could peel out one thread from the rope, we could have one ascending neural pathway from the receptor up through the cord towards the control center of the brain. And in that one thread that helped make up the rope of the peripheral nervous system nerve and the central nervous system tract, if we could pull out that one thread, we would see that it contained three neurons: a first order or primary neuron, a secondary neuron, and a tertiary neuron. The first order or primary neuron is connected to the receptor and carries that information about the stimulus from the receptor to the central nervous system. The secondary neuron is actually an interneuron. So it's going to connect that primary neuron to a tertiary neuron. And the tertiary neuron, which is also an interneuron, is going to carry the signal from the secondary neuron up to the cerebrum in the brain. We looked at this drawing of this thread that travels from the peripheral nervous system at the site of the stimulus all the way up to the cerebral cortex in the central nervous system. Here's that primary neuron shown in blue. Imagine, depending upon where the stimulus was applied out in the peripheral nervous system of the body, this axon that connects the receptor to the secondary neuron, this axon could be very, very long. Some axons are very long in these afferent or sensory neural pathways. The primary neuron is going to synapse with the secondary neuron here. The secondary neuron is an interneuron. It connects the primary in blue with the tertiary neuron in yellow. This tertiary neuron is going to carry the information from the secondary neuron to the cerebral cortex. And this drawing is just another way of visualizing the same pathway. It's just one thread, one sensory pathway. In reality, there would be multiple copies of this thread, all bundled together to create a nerve in the peripheral nervous system and a tract in the central nervous system. But we've pulled one thread off of that rope. The primary neuron in this sensory pathway carries the information from the receptor to the central nervous system. The secondary neuron, which is an interneuron, carries the information from the primary neuron to the tertiary neuron. And the tertiary neuron carries it up to some cell in the brain that happens to be located in the cerebral cortices. Of course, we could also pull a thread out of the rope that makes up descending or motor tracts. We could take a tract and peel it out of the central nervous system and keep its connection to some peripheral nerve. And if we peeled one thread out of that long rope, we wouldn't see 3 neurons this time, we would see 2. An upper motor neuron and a lower motor neuron. The upper motor neuron is going to have its cell body in the cortex, or perhaps the brain stem. So there are 2 locations where motor neural pathways can begin, either in the cerebral cortex itself or in the brainstem. If the upper motor neuron begins in the cerebral cortex, it's going to synapse with the lower motor neuron in the brainstem. If it begins in the brainstem, it's going to synapse in the spinal cord. Whichever the case, it's going to synapse with the lower motor neuron, which is then going to travel out and terminate at the effector structure, either the muscle or the gland. And that's what this picture demonstrates for us. There are 2 neurons in a motor neural pathway. There's an upper motor neuron and a lower motor neuron. The upper motor neuron sometimes originates in the cerebral cortex and sometimes in the brainstem. It's going to synapse with a lower motor neuron who has its neuron cell body either in the brainstem or in the spinal cord. That lower motor neuron is then going to send information out into the peripheral nervous system, where it will synapse with the effector structure, whatever that happens to be. At this point in the lecture, we left behind the spinal cord and started talking about the spinal nerves. Remember, a spinal nerve is in the peripheral nervous system because we've left the central nervous system. The central nervous system includes the brain and the spinal cord. Everything else is part of the peripheral nervous system. There's some terminology here that can be confusing, particularly when we talk about roots versus horns. There's a dorsal root and a ventral root of each spinal nerve. The dorsal and ventral roots come off of the spinal cord and combine together to make the spinal nerve. The horns, the dorsal and ventral horns, are the gray matter within the spinal cord. So be careful, don't get those terms mixed up. Remember, the dorsal roots carry sensory information and are associated with a ganglion, the dorsal ganglion. The ventral roots are different. They're carrying motor information. Once the dorsal root and the ventral root come together, they merge into a single spinal nerve. Spinal nerves, remember, are paired. So for each spinal nerve, there are really two. There's one coming off on the left and one coming off on the right. And because spinal nerves are a combination of sensory and motor fibers, we call them mixed nerves. They contain both sensory and motor information in them. Remember, it turns out that most nerves are mixed. Spinal nerves are mixed, and most other nerves in the peripheral nervous system are mixed. When we look at the spinal cord in cross-section, these are the horns. Dorsal horn, ventral horn. Dorsal horn, ventral horn. What's coming out of the spinal cord, these are dorsal root, ventral root, and they're going to combine into a spinal nerve. Over here on the right side, we see the same thing. Dorsal horn gives rise to the dorsal root and the dorsal ganglion, which carries sensory information. The ventral horn gives rise to the ventral root, which carries motor information. These two are going to combine into a spinal nerve. It's these spinal nerves that are then going to branch and branch and branch some more and give rise to all of the peripheral nerves that exist. So all of the nerves in the peripheral nervous system are going to come into or out of these spinal nerves. As soon as the dorsal and ventral roots come together and form a spinal nerve, that spinal nerve is going to exit the vertebral column. It actually passes out through the intervertebral foramen and starts to branch right away. And we talked about these four primary branches. The anterior branch, or ramus as it's called, the posterior ramus, the meningeal branch, and the communicating branch. That meningeal branch is going to turn right around and re-enter the vertebral canal so they can innervate things around the spinal cord, including the meninges, the vertebrae themselves, and spinal ligaments. The posterior ramus is going to innervate muscles and joints along the midline of the back and the medial skin of the back. The anterior ramus innervates the lateral musculature of the back, the lateral skin of the back, and gives rise to the nerve plexes. So this anterior ramus is the one that branches quite extensively. The other one that branches quite extensively is the communicating branch, because it's going to end up innervating the viscera. And this is the image that we looked at to help us differentiate what is a root, what is a spinal nerve, and where these branches come from. Here is the spinal cord in cross-section. We can see a dorsal root coming off here with a dorsal ganglion and a ventral root coming off. Those two are going to join and produce a spinal nerve. Once the spinal nerve exits the vertebral canal, the bone of the vertebral canal, it's going to start branching. Here's that meningeal branch that's going to go back towards the spinal cord. Here's the dorsal branch. Remember, that also translates as the posterior ramus. Here's the ventral branch, or the anterior ramus. And here's that communicating branch. We said that the anterior ramus actually branches quite extensively. So we talked about it in a little bit more detail because it's the anterior ramus that comes off of each spinal nerve that is going to create what are called the nerve plexes. There are actually 5 of these. There's a cervical plexus, a brachial plexus. Remember, there's no thoracic plexus. There's a lumbar plexus, there's a sacral plexus, and there's a coccygeal plexus. All a plexus is a big tangled web of nerves. So up in the area of your neck, you have a cervical plexus, a big tangled mass of nerves that are coming off of the anterior ramus in that region and innervating structures in your neck. The brachial plexus is coming off in the region of your underarm, basically, and innervating structures in your arms. You've got a lumbar plexus and a sacral plexus that are sending out nerves into your legs, and of course a coccygeal plexus as well. These plexi or plexuses, again, are real meshworks or bundles, branching bundles of nerves. You can imagine that it can cause a whole lot of harm to someone if one of these plexuses gets damaged. There's a lot of nerves that are inside a plexus, and so if a plexus gets damaged through traumatic injury, it can cause some very severe effects. This was the image that we used in lecture just to demonstrate the complexity of what a plexus is. Remember, that anterior branch of each spinal nerve is going to give rise to these various plexuses: cervical, brachial, lumbar, sacral, and coccygeal. The last thing we talked about in this lecture was the dermatomes. There is a map, remember, of all the dermatomes on the human body. Each of the spinal nerves, with the exception of C1 spinal nerve, receives sensory input from very specific regions of the skin. And each one of these regions of the skin is called a dermatome. Of course, each spinal nerve receives sensory information from other areas as well. But we're interested in which areas of the skin each spinal nerve is connected to, because we can touch the skin. We can touch the skin with a pin, and we can know that if the person can feel the pin in that area of the skin, it means that that spinal nerve is intact, it's working. If you were to touch a pin to an area of the skin and the person didn't feel it, that would tell you that there was a problem with that particular spinal nerve. Now, these dermatomes, these areas of skin that are connected to each individual spinal nerve, these dermatomes are mapped. And these maps are readily available for physicians. So if a physician suspects, for example, that there might be injury to, let's say, the T3 spinal nerve, the physician can use a pin to prick the skin in the region of skin, the dermatome, that is connected to the T3 spinal nerve. If the patient can't feel the pin, you've got a problem. These dermatomes are very helpful in medicine for tracking injury, sensory information that's related to individual spinal nerves. That's the end of Monday Minutes for this week. Remember, if you have any questions about the material that we've reviewed, be sure to message me through Canvas.